So you have seen the aerobic respiration in germinating seeds. Now aerobic respiration in green plants. This is the aim. Experiment to demonstrate that aerobic respiration in green plants. This is the experiment, aim of the experiment. All right. Now, what we will do here, we make an arrangement here. And arrangement, we take it out. We have taken the green plant and put it in the well jar. All right. Now here we have taken the beaker. Cork is put it here and here also. All right. Now this is, in this we have the lime water and its name we have given lime water A, and this is lime water B. This is a green plant, this is bell jar. And here the lime water B. Fitted with lime water. Got it clear? Now all these has to be connected. So in order to connect here, we put some hole here, cork with the help of hole here. All right. Now, here is it visible there? Yes. Here we have put soda lime. Soda lime. And it is connected from outside in this side. And then with the help of second cord, we connect this here. And another delivery tube. We are connecting it here with the beaker T. In this way, all these are connected. All right. Now, here we have the suction or aspiration pump. suction or aspiration pump. This is the setup. Got it clear? Now the experiment setup as shown in the diagram. Method. You have to directly make the setup and say in the method the experiment setup as shown in the diagram. Alright. Now this is the suction or the aspiration pump. Alright. So when the aspiration pump or the suction pump opens, or you suck or push this aspiration pump, so what observation you will get and why? So what you will see, air rushes from here because it is outside. This is outside. Air from outside will come here, passing through the soda line, then the beaker A, then the plant, then the beaker B. As soon as you push this aspiration pump, air from outside rushes, passing through soda line, rushes line water A, line water B. What will you observe? Aim, remember, aerobic respiration in green plants. Aerobic respiration in green plants. Now, what will be your observation? Yes, anyone can give this observation? Anyone? All right. Write the observation there. Lime water A does not turn milky, but lime water B turns milky. Lime water A does not turn milky, but lime water B turns milky. What will be the reason for observation? Why lime water A does not turn milky? Why lime water B turns milky? Reason for observation. See, look at here. The air is coming from outside containing oxygen and carbon dioxide. Is it true or no? Yes. But soda lime here, it is the absorber of carbon dioxide. So it will absorb, all right? It will absorb carbon dioxide. So air is a mixture. So oxygen and carbon dioxide is here. Carbon dioxide will be absorbed by this soda lime. Now here only the oxygen will come. Oxygen will not turn lime water milky. Then this oxygen passes from here to the green plant. This green plant will respire, take in oxygen, producing carbon dioxide. That carbon dioxide will go from here and carbon dioxide turns lime water milky. Got it clear? The reason for observation, right? It lime water it does not turn milky because the air is free from carbon dioxide, because soda lime absorbs carbon dioxide of the air, and therefore air is free from carbon dioxide. Whereas lime water B turns milky because air contains carbon dioxide. The green plants consume oxygen of air and produce carbon dioxide during respiration, which turns lime water milky. So you have to mention it here. air is free from carbon dioxide here because soda lime absorb this carbon dioxide. So lime water does not turn milky, but oxygen reaches here. Green plants respires with the help of this oxygen, produce carbon dioxide, which passes through the lime water bean and turns lime water milky. Conclusion. The experiment. The conclusion of this experiment is that green plants respires aerobically, or aerobic respiration takes place in green plants, that is taking oxygen or consuming oxygen and producing carbon dioxide. That will become the conclusion of the experiment. All right. Now, questions. Council has asked you the question on the basis of this. The first question, write your observation. The setup they will give you. Write your observation. So you will write your observation. All right. Lime water A does not turn milky. Lime water B turns milky. You will have to see the situation in the diagram. Here, the suction aspiration pump is this side. Air is coming from this side. So you have to see. Anyone can just reverse it. They put suction pump here and air this side. All right. Give reason for your observation. Now name the process demonstrated. Which process will you define here? Which process? Name the process demonstrated. Hurry up fast. I want the answer. Yes, Pratik. Unmute yourself, Pratik.
Sir, respiration, aerobic respiration.
Respiration, but here you will go for aerobic respiration. All right. Because taking in oxygen and giving out carbon dioxide. Aerobic respiration where oxygen is consumed and giving out carbon dioxide. So name the process demonstrated. So this will be aerobic respiration. Define the process, you will define it. Give an overall equation of this process. You can give the overall equation of this process. Next question. What is soda lime? And what is the purpose of soda lime? So important question here. What is soda lime? And what is the purpose of soda lime? Write it there. Soda lime, it is a mixture of sodium hydroxide and calcium oxide. You will write in word. Soda lime. It is a mixture of sodium hydroxide and calcium oxide. Calcium oxide is quicklime. Okay. The soda lime, it is a mixture of sodium hydroxide and calcium oxide. What is soda lime? And what is the purpose of soda lime? The purpose of soda lime is to absorb carbon dioxide of the air. Purpose of soda lime is to absorb CO2 from air and make the air free from carbon dioxide. And make the air free from carbon dioxide. Got it clear? Rest. What is the control of the experiment? So the same experimental setup. What will be the control of this experiment? The same experimental setup, but in place of green plant, take a wooden stick. So the same experimental setup, but with a wooden stick. This will become the control of the experiment. Now, no one has asked me, sir, why you have put the bell jar here. You can directly do it here, na? Green plants, you put it here. Why we are putting here the bell jar? Yes, Alankrita.
So we put the bell jar because if we won't, then all the oxygen will escape in the air and carbon
All
dioxide.
right,
It will get mixed up, and air already
mixed
has
up.
it.
Okay,
So the experiment will not work then without bell jar.
if we will not put the bell jar, what will happen here?
We will not be able to
You know,
prove that.
this plant will go for photosynthesis, and photosynthesis, it will consume the carbon dioxide and produce oxygen. All the carbon dioxide will be consumed here, so how it will reach here? Got my point? To write it here. Why is that? To write the question also, this is also important question. All these questions are from the board only. Alright, earlier this chapter was included in class 10. So they ask you. And similarly, they ask you in class 9 in this way only. To write it there. Why is the bell jar covered? Why is the green plant is covered with bell jar? To stop photosynthesis in green plants. Write the answer to stop photosynthesis in green plants. Alright? Alright. So next experiment, what we have, experiment number 4. All these are the questions which is important one and you have to plan it thoroughly. Now, in experiment number 4, to demonstrate that anaerobic respiration in germinating seeds. Now, we go for aim of the experiment as anaerobic respiration. In germinating seeds now, not in green plants, in germinating seeds. Got it clear? Now, in this, take a test tube and place some germinating soaked seed with disinfectant. Nothing, you're doing it. Test tube. TD, test tube. Putting some germinating seed. Cotton wool. So take a test tube and place some germinating seed soaked with disinfectant. You know the reason for soaked in disinfectant? To kill the bacteria. Inside the test tube filled with mercury. Inside this test tube, you will fill with mercury. So this is mercury. This is germinating seeds soaked, soaked in disinfectant. Right. This is cotton wool plugging. So that is tube filled with mercury. Now invert, place this test tube inverted in a glass tub containing mercury. So first we will make a glass tub here. This glass tub also containing mercury. Now this test tube you will invert like this. All these will become in this way. In this way. Germinating seed will be here. So you put it inverted. Germinating seed. Filled with mercury. Cotton wool. Method में take a test tube having germinating seed soaked with disinfectant and with the help of cotton wool plugging add mercury in the test tube. Invert the test tube, put in the trough containing mercury and observe after some time. Observation. What will you observe? Yes. Anyone? Write your observation here. The level of mercury. Observation. The level of mercury in the test tube. This is the mercury in the test tube, and the level of the mercury decreases. Observation में the level of the mercury in the test tube decreases, falls, and colorless gas collects at the bottom of the test tube. So here the colorless gas will collect at the bottom of the test tube. Just now I am not saying it is a carbon dioxide, but it will be a carbon dioxide. But in observation, I am writing it here. The colorless gas collects at the bottom of the test tube. Since test tube is inverted, now this will become the bottom. Level of mercury decreases and the colorless gas is collected at the bottom of the test tube. This is carbon dioxide, but just now in observation we have not tested. So you will mention colorless gas collected at the bottom of the test tube. Now conformation of the gas evolves. Conformation. Next heading, conformation of the gas evolves. Take some pellets of potassium hydroxide. Pellets of, here you will use potassium hydroxide pellets. Take some pellets of potassium hydroxide and put it into the stuff. Now this potassium hydroxide, in the bracket right, it is an absorbent of carbon dioxide. So what you will do, you take the pellets of potassium hydroxide and put it in the trough. What will you observe? The level of mercury in the test tube increases. The level of mercury in the test tube increases. It means it absorbs all the carbon dioxide. That's why level increases. Take some pellet of potassium hydroxide, which is an absorbent of carbon dioxide, and put in the glass tub. The level of mercury in the test tube rises as the gas of the test tube is absorbed by KOH pellets. And since KOH is the absorbent of carbon dioxide, therefore it confirms that this gas is carbon dioxide. Got it clear? Again, I am repeating. Take some pellets of potassium hydroxide, which is an absorbent of carbon dioxide. Put it in the glass tub. The level of mercury in the test tube rises as the gas of the test tube is absorbed by KOH. Since KOH is the absorbent of carbon dioxide, This confirms that this gas is carbon dioxide. Got it clear? Now, reason for observation. This germinating seed respires anaerobically. Germinating seed respires anaerobically. Why the gas is collected here? Because the germinating seed respires anaerobically and produces carbon dioxide, which is collected at the bottom of the test tube. That you will write for the reason for observation. Write it. Germinating seed respires anaerobically and release carbon dioxide, which collects at the bottom of the test tube. Which collects at the bottom of the test tube, displacing, this word is very important, displacing the mercury downwards. Write it, hurry fast. Germinating seeds respire anaerobically and release carbon dioxide, which collects at the bottom of the test tube, displacing the mercury downwards. All right. So this is the setup, the way you will go for the answer. Control of the experiment, the same experimental setup, but with the dead seed, or we say the boiled seed. Either you take the dead seed or the boiled seed, no observation will take place. That will become the control of the experiment. All right. All these are the experiments which you have to go through in this chapter. These are the experiments which are in your course, which you have to go through. And now this chapter is also completed. All right. I have already told you the differences between the photosynthesis and respiration there. Now, photosynthesis, it is a catabolic process. Respiration, it is an anabolic process. All right. Photosynthesis is an anabolic process, sorry, and respiration, it is a catabolic process. Now, photosynthesis may oxygen is released, carbon dioxide is used up. Respiration, oxygen is consumed and carbon dioxide is given out. These are the both are antagonistic process, opposite process to each other. The difference is given on page number 60, table 7.2, photosynthesis and respiration. So these are the differences which we have already discussed in the earlier only, while I was teaching you anabolic and catabolic processes there. All right. To go through the chapter thoroughly once again. Then the last point, they have given respiration in plant, which is compared with the respiration in animal. So in plant, there is no gaseous transport. They have compared the respiration in plant and respiration in animal. So respiration in plant, there is no gaseous transport. Transportation now takes place, like in our body. Transport of oxygen takes place through the blood. Transport of carbon dioxide takes place through the blood. So no gaseous transport is there in the plants. All right. But in animals, yes, it is there. Blood is doing the transport of respiratory gases, that is oxygen and carbon dioxide, transport of oxygen from lungs to body tissues, and transport of CO2 from body tissues to lungs. This is done by blood in respiration in animals. Secondly, one of the end products of anaerobic respiration in plants is ethanol or ethyl alcohol, while in animal it is lactic acid. In ourselves, we have the lactic acid, but in plant, ethyl alcohol. That also I have discussed. Thirdly, in plant, the process produces a little heat as compared to animal. We have a lot of heat produces in our body. In that way, our body temperature is maintained. All right. So more heat energy is produced in our body. In this way, we have come to this end of this chapter. Now, question answers and the review questions are is given on page number 61. Go through the chapter, read it thoroughly, learn it, and then we will discuss the question answer. All right.